Welcome all of you to the latest installment of the SLAC Public Lectures. We're very glad that you can make it. We're sorry that we're still in this remote mode, but we'll see how that lasts and whether we can be in person in the new year. So it all remains to be seen whether there's a fifth wave or not. For the moment, we've got a great lecture today. Photosynthesis is probably the most important catalyzed reaction in nature. It is literally the way that an uninhabitable planet was turned into the planet with the oxygen-rich atmosphere that we're all breathing right now. And how it works has been a mystery for a long time. It's a very complicated process to create oxygen from ordinary organic materials. Franklin Fuller has been studying this process basically his whole career. He was a PhD at the University of Michigan and basically studied photosynthesis using lasers and chemistry. He then went on to a postdoctoral fellowship in Berkeley at the Lawrence Berkeley Lab, and there started using X-ray spectroscopy to understand how this process works. In 2017, he came to SLAC as one of our Panofsky Fellows. And here he's been developing really very sophisticated X-ray techniques to get at the various steps in the photosynthetic reaction, in particular in the half of the chain called Photosystem II. And we're very glad to have him here today to explain what he's learned in this process and to teach you how photosynthesis works atom by atom. So Franklin, please take it over.
All right, thanks, Michael, for that great introduction. All righty, so why do we need oxygen to live? The reason that we need oxygen is because it's what allows us to extract energy from the food that we eat. And so in every one of our cells, there's this reaction that depends on oxygen, which is going on. And this reaction is also found inside of internal combustion engines, like, for example, in your automobile. In this reaction, we are taking a carbon-based fuel like sugar for ourselves, or in the case of a car, gasoline, and mixing that with oxygen and getting them to react. And when they react, they will form a mixture of carbon dioxide and water, and we of course breathe out carbon dioxide, and so does carbon dioxide also comes out of the exhaust out of automobiles. And this is happening because the carbon atoms in the fuel are bonding to the oxygen atoms in the air that we breathe in, and they form CO2, and the hydrogen atoms in the fuel mix with the oxygen atoms of the O2 gas, and that forms water. So on the right-hand side of this equation, we have CO2 gas and water, and the chemical potential energy of these products is much lower than the chemical potential energy of the thing that went into this reaction. And that means that this reaction is going to release a lot of energy. Now, of course, in internal combustion, this energy is released very rapidly and violently as a bunch of heat and pressure, and that pushes on a piston to drive the engine. In our cells, this reaction is more controlled, slower, and the energy from this reaction is harvested into a sequence of intermediate chemicals. Those chemicals then float around inside of our cells and deliver energy to various parts of the cell so that we can do things like move our muscles and so on. Now, if you were to wonder where is the energy coming from in this reaction, I think it's normal to think that the energy is coming from the fuel, and that's not incorrect. There is energy that results from breaking apart this fuel molecule, but actually a large portion of the energy in this reaction is supplied by the oxygen gas itself. And that's pretty interesting when you think about it, because of course we live on this planet, as Michael just said, that's got nearly 20% of the atmosphere made of oxygen. And so what that means is that we are sort of in a giant chemical potential energy fuel tank where we can just breathe in chemical energy on demand. And this is extremely convenient, of course, for us animals and our cars and our airplanes and so on, because this means that we don't need to carry oxygen around with us. Imagine if we had to, in addition to the fat stores we have in our body, we had to carry around sacks of oxygen in order to carry out this reaction. We would be much heavier and slower. And for example, this is why airplanes use jet engines, because jet engines can take advantage of the oxygen that's in the atmosphere and to produce thrust. If we instead used rockets to power our airplanes, we would have much less cargo capacity because we would have to spend the weight for that amount of thrust on storing and holding onto the oxygen. So all this is to say that oxygen is really great. It's really wonderful and convenient that we have so much of it in the atmosphere, and this is enabling our life as animals and as the technological products that we use today. And of course, as Michael said, oxygen comes from plants. That's what has put all this oxygen into the atmosphere. And the process which produces it is called photosynthesis. Photosynthesis is a complicated process. It's not just a single reaction, and it's got two main parts. I think one of the most famous parts of photosynthesis is the fact that plants will take in carbon dioxide from the atmosphere, and by spending some of the cellular energy that they've built up, they can turn those carbon dioxide atoms or molecules into larger carbon molecules like sugars and starches and lignins and so on. And this process is known as carbon fixation because the plant is taking carbon dioxide out of the atmosphere and turning it into a solid material like wood or plant material. Now, the cellular energy that is needed to power that carbon fixation portion of the reaction, that's coming from the solar energy conversion half of photosynthesis. And that half, light is harvested and turned into chemical energy in some way that I'll be explaining in a few slides, so that it can power this carbon fixation process. The energy harvesting and conversion portion of photosynthesis is mainly done in the green parts of plants. So for example, a leaf, and if you zoom in on that leaf down to the cellular level, we have organelles called chloroplasts. Chloroplasts are kind of like mitochondria of the cell. And if you're around my age, you'll know of mitochondria from The Fresh Prince of Bel-Air. Mitochondria is the powerhouse of the cell, and chloroplasts are the powerhouse of plant cells. So inside these chloroplasts, there's all sorts of weird structures going on in here, and it depends on the species of plant how this thing is structured. So if you keep zooming in to the point where we reach this thing that's highlighted here, is called the thylakoid. That's where the proteins are held, and they're held in these little membranes, so film bubble-like materials that hold on to singular proteins. And at that level, which is what I'm depicting here, we can do individual reactions that comprise the overall photosynthetic process. So the reaction which is producing oxygen that is in the atmosphere is called the water oxidation reaction. And it's done by a protein called Photosystem II. And this is the overall reaction scheme up here. We start with 2 water molecules, and with some energy input in the form of 4 photons, we produce oxygen gas. And 4 hydrogen ions, also known as protons, and 4 electrons. So what happens in this reaction is that oxygen is actually a waste product. The plant just throws it out, which is surprising when you think about it, because I've just argued that oxygen is this great resource of chemical energy. And so you may wonder, how is it that plants can afford to throw such a valuable resource out? And the answer is that they can afford to do this because they do still get something out of the reaction. Those 4 hydrogen ions and the 4 electrons there, those are a raw form of chemical energy that the plant can keep and use. And when you look at the left-hand side of this equation, you'll see that they don't actually have to pay that much to get that raw form of chemical energy. All they have to do is be on a planet where there's a lot of water and sunlight. So for example, in the oceans where this protein first evolved, of course, there's plenty of water around and plenty of sunlight at the surface of the ocean. So the fact that they give off oxygen and throw it away doesn't matter, because they're basically, it's like free real estate, all this water and all this this sunlight. Now, you may not be familiar with water oxidation, but you probably are familiar with the reaction that's related. It's called water electrolysis. Now, this reaction is something you can do at home or probably have done in a school lab. You take a 9-volt battery and hook up the 2 leads of the battery and dip them into water. And what will happen is that oxygen gas is going to form at one of the electrodes, and hydrogen gas is going to form at the other electrode. And what's happening at the oxygen forming electrode is the water oxidation reaction. What's happening at the hydrogen forming electrode is that the hydrogen ions formed in the water oxidation side mix with electrons, and they recombine into hydrogen gas. So photosystem II doesn't want that second part of the electrolysis reaction to happen. It only wants the water oxidation reaction to happen, because the hydrogen ions, as I said, are a raw form of chemical energy. Think of hydrogen ions and electrons as gold ore for chemical energy. Gold ore is valuable, but you can't go to the store and buy something with it. So what the plant needs to do almost immediately after it gets these 2 things, the hydrogen ions and the electrons, is it needs to turn them into something more fungible, like cash. So the cash of the cell in this case is the hydrogen ions are converted to what's called ATP, which I think is a more famous molecule. And so we can call that the dollar bill of the cell in terms of transporting chemical energy. And then the electrons, they go on the other side of the membrane, and those end up over towards photosystem I, where we form a molecule called NADPH. It's also present in our cells. It's another way to transport chemical energy around. And so let's just call that the euro of the cell. And if you have enough euros and dollars in hand with carbon dioxide, then the plant can spend that to produce things like sugar. And that's how this all hooks up to the carbon fixation portion of photosynthesis. So by this point, I think it's clear that photosystem II is important. It's what is producing oxygen that's in our atmosphere, but that doesn't really convey the totality of the importance of photosystem II until you understand how long it's been around. So geologists have used a variety of techniques to study the history of how oxygen was put into the atmosphere and how much has been in the atmosphere, using a variety of techniques, looking at dissolved oxygen levels in ice and iron oxide deposits throughout the strata of rock. And they've come up with a model that looks sort of like this, where if you look almost 4 billion years ago, you'll see that the oxygen in the atmosphere at that time was very low, less than 1% oxygen. And by the time we get to 3 billion years ago, evolution came up with a way to take advantage of this water and sunlight abundance and to turn it into chemical energy. And that's when oxygenic photosynthesis was born. We have to wait nearly 1 billion years later for the arrival of aerobic metabolism. That's what we do. That's the process where we use oxygen to extract energy from carbon-based fuel sources. And so in that time, what was happening is that the oxygenic photosynthetic organisms were pumping out oxygen in the atmosphere. And only when there was enough oxygen in the atmosphere to take advantage of it did aerobic metabolism arise. And this is also coinciding with something that you may have heard of, which is called the Cambrian explosion. At this time, there was this huge increase in the diversity of life and multicellular life. And so it's clear that oxygenic photosynthesis is important for really spawning the life that we know of today. What's even more interesting is that if you use genetic dating techniques where they look at the rate of change of genes over the course of time, you can put a date on how long ago it was that photosystem II as we see it today, how long ago was that produced in the evolutionary chain. It turns out that photosystem II, as we know it today, is basically the same as it was 3 billion years ago, which is crazy. This protein is like an eldritch monstrosity. It is extremely ancient. And the portion of photosystem II, which is producing the oxygen, is largely, or not largely, almost completely preserved from 3 billion years ago to today. And what that means is that photosystem II is the best that nature could come up with in terms of taking advantage of this water oxidation reaction. You would think that if there was a better way to do it, that would win out in the course of 3 billion years. And so far, there has not been a different way to do it. So this means that photosystem II is extremely interesting. It's a pillar of biology, really. Okay, so let's have a quick look and see what we're talking about when we talk about photosystem II. This is a structure of the photosystem II protein, and I'll talk in a moment about how we are able to produce an image like this. But anyway, so photosystem II comes usually as a 2-pack. You get these 2 halves of the system are the same, and they're stuck together, and so that's convenient for displaying it. On the left side here, I've got the internal core of photosystem II on display, and at the top we have some quinones. They transport the electrons out. In the middle we have chlorophyll, which I'm sure you're familiar with. That's what makes plants green, and they absorb the light, and that the energy from that light absorption event is then transmitted down to this thing called the oxygen evolving complex, which I'm showing at the bottom. And that is made out of 4 manganese atoms, 5 oxygens, and 1 calcium. And this whole thing is wrapped inside of a very complicated protein structure, which is what I'm depicting on the right side here. The purpose of this structure is to hold it in the membrane. So that's what these dotted lines are. They depict where the membrane is, and these coils anchor it in that membrane. And part of it hangs out on the bottom side here, the lumen side, so to say, of the thylakoid membrane. So this is the photosystem II picture, and because of how important photosystem II, it's been studied by many researchers for many decades. And I myself got into the business, as Michael said, around 12 years ago, where I was studying just the early light absorption event in photosynthesis in the chlorophylls that I was mentioning earlier. So in that case, I was using what's called multidimensional optical spectroscopy to study the very early time events that lead up to the generation of an electron after light is absorbed. This is a screen capture of a YouTube video where I'm giving a tour of our laser spectroscopy setup for that experiment. What I want to talk about today, though, is the business end of the oxygen evolving complex, or of photosystem II, which is the oxygen evolving complex. And that's where oxygen is being produced, and where the hydrogen ions and electrons are being extracted and giving chemical energy to the cell. And so to do that, we're going to use 2 different techniques, one called X-ray crystallography and one called X-ray emission spectroscopy. And we've done these 2 techniques here at SLAC mainly using a unique X-ray tool called the Linac Coherent Light Source, which is an X-ray free electron laser. I'll explain that a little bit more in detail in a few slides. But first, I want to talk about these 2 methods, X-ray crystallography and X-ray emission spectroscopy, and how they help us understand this photosynthetic process a bit better. So one of the main goals that we want in order to understand photosystem II is we want to have an atomic level picture of what's going on. So that slide I just showed you of the whole structure, that was created using this technique called X-ray crystallography. And this technique is really important because it gives us the ability to see where every single atom is in this protein to a resolution of down to 200, or down to 2 microns, or 2 angstroms. 2 angstroms. A hydrogen atom is about 1 angstrom in size. So if you are able to resolve down to 2 angstroms, you can't quite see hydrogen atoms individually, but you can see just about any other atom, especially oxygen. So when we want to know where waters are, being able to achieve a 2 angstrom resolution is adequate for us to be able to say precisely where every single water is. So this technique of crystallography is a complicated technique and to explain, but basically what we need is we need to provide a protein crystal. What that means is we need to take PS2, the photosystem II complex, purify it to a very high level of purity, and through some wizardry of chemistry, turn it into a crystal. What that means is we're going to have every one of these photosystem II atoms or molecules lined up next to each other in a row and row on row until it forms a sheet, and sheet on sheet until it forms this dense packed cube or whatever structure it wants to form of photosystem II molecules all lined up with each other. And the more perfect that lattice is and the more pure the PS2 is, the better this technique is going to work. Once you have that crystal, you can hit it with an X-ray beam like we're showing here, and it will form these just frankly gorgeous patterns of spots on the detector in front of it. And these patterns, they reflect properties of or they don't reflect, they indicate properties of the lattice structure, while the relative intensity of these spots, that's what encodes the atomic positions of the individual atoms that are creating this lattice structure. So by rotating the crystal around and collecting these different patterns of spots, we can infer the atomic position of all the atoms in the molecule. Okay, so these are what protein crystals look like. They're little tiny things here. These are about 30 microns in size, and a micron, 100 microns is about the width of a human hair. So these are maybe a third the size of a human hair across. And making these things is a very difficult task. And so over the years, I'm showing a sequence of publications where we were looking at the OEC, where the water oxidation reaction is happening, and showing the progress that we've made in terms of resolving it. In 2012, we were at like 6.5 angstroms, and as we progressed onward to the current day, we're down to about 2 angstrom resolution. And at this point, we can finally start to clearly resolve these oxygens from their other atoms. And I mentioned the other technique that we used is X-ray spectroscopy. X-ray spectroscopy is it's basically, it measures, X-ray emission spectroscopy is a measurement of X-ray fluorescence. And fluorescence, you may be familiar with, things under black lights that glow, essentially like the security stripe on this $20 bill. The idea is that there's a molecule, in the case of X-rays, it'd be an atom that absorbs a photon of light and then emits a photon of light at a different color. So these atoms in this $20 bill are absorbing in the UV, you can't see it, and then it emits it out in the green light region, which is lower in energy. And the same is true for X-ray fluorescence. It absorbs some high energy X-ray photon, and then depending on which element absorbs it, it will emit an X-ray photon of a different color, and that color is specific to the element that is emitting it. So here I'm showing all these different lines. These are showing where different elements emit. And for example, we'll be able to use this to see manganese and separate it from iron or oxygen or carbon or any of the other atoms that are in the molecule. If you zoomed in on one of those spikes in that graph I just showed you, then you would be able to see things like whether or not a manganese atom has gained or lost an electron. So in this graph, I'm showing a manganese 2, which means it's missing 2 electrons from its outer shell, and then there's a manganese 4 atom, which is missing 4 electrons. And so by looking at this peak shift, we can tell how many electrons are lost in this manganese atom. That's going to be important for how we can track the reaction of photosystem II. Okay, so as I mentioned, many researchers have been investigating photosystem II for many years, and specifically, they've been investigating the water oxidation reaction for many years. As early as the 1970s, we have this picture of how the reaction cycle works. And it's kind of like a 4-stroke engine situation where there are 4 different light absorption events. You know, there are 4 photons that were required to do this reaction, and they are absorbed in sequence. And every time an absorption of a photon occurs, something happens in the protein, and it advances the state of the reaction cycle. What they found was if you take a flash bulb, like a photography flash bulb, and you flash solutions of Photosystem II, and you do this in sequence, you will be able to measure the oxygen yield per flash, and it gives us this cyclic pattern of oxygen yield. You go 4 flashes here, and then the next set of 4 flashes will release a ton of oxygen, and so on. And it's because they're now synchronized by that flash system. And so they came up with this cycle scheme to describe that. And then subsequent to the 1970s, people have been investigating this cycle for some time. In the mid 80s, they were able to make a very sensitive pH probe that could detect that between these flashes, certain ones between S2 and S3, S3 and S4, and S0 and S1, that hydrogen ions are being released. And in the mid 90s, they used different spectroscopies, including X-ray spectroscopies, that would show when electrons are being released in the cycle. And they also found that waters are being absorbed at specific steps, specifically between S2 and S3 and S3 and S4. There are 2 waters that come in, and they used isotope studies and mass spectrometry to determine when the water was being taken in and what part of this reaction cycle. So quite a lot about this reaction is known, but the atomic level of detail is missing here. We don't know where the water is bound to. We just know that it is bound in this step. We don't know how those hydrogen ions are being transported out. We do have some indication of how the electron is being transported out, and I'll indicate that later, but there's a lot of open questions about how this reaction works at this stage. So now that we have a picture of what we want of that reaction cycle, what I want to do is talk about our experiment where we've investigated the S2 to S3 transition. If you want to know what happens or how oxygen is created, you might want to look at S4, where oxygen is actually released. But this is hard to examine because S4 is a very transient state. And so we picked this setup transition to look at, S2 to S3 transition. Because if we looked just before and just after the oxygen is released, we should be able to make a good inference about how oxygen was created in this S4 step. So our experiment that we're going to do is to look at this S2 to S3 transition, and we're going to finally resolve it in time using X-ray crystallography as a probe and X-ray spectroscopy as a probe to watch how the atoms are moving and how the electrons are moving in this transition. And we're going to do this with microsecond resolution. Microsecond is a millionth of a second. These transitions can take milliseconds, which is a thousandth of a second. And the fastest that Photosystem II can cycle around is around 500 times a second. So by using microsecond steps between S2 and S3, we should be able to get a good idea of how this transition is going to take place. Okay, so I mentioned earlier that we use a tool called the X-ray free electron laser. And it's an interesting tool because it allows us to study these reactions at room temperature, which is unique for X-ray science because normally the X-ray pulse is so long in time that if you expose the sample too much, you will damage the sample. So you know, of course, that radiation is bad for you. X-ray radiation is especially bad for you, and it's going to destroy whatever you're looking at if you hit it with too much radiation. But we want to hit it with a lot of radiation because we need a lot of signal to noise to be able to resolve these small changes. And how XFELs help us do this is by providing an extremely short pulse, a pulse that is so short that on the timescale of the duration of the pulse, atoms are completely frozen in time and electrons are moving relatively slowly. And that time scale is called a femtosecond, and XFEL pulses around 10 femtoseconds long. And it's an astronomically short duration. We're talking 1 femtosecond is a millionth of a billionth of a second. So that's what's unique about XFELs and why we use them, because it allows us to watch this reaction at room temperature in real time. Oh, and the important thing was that because we're able to put so much dose into this sample, we do end up having to pay the Pied Piper in the sense that we will destroy the sample in a single shot, but it takes about a microsecond for that sample to blow up. And by this time, the signal that we wanted was collected and done, and everything was completed. And then later on, a coulombic explosion will occur and the sample will destroy. And that's what we're showing here. So it's actually a stroboscopic view of the sample coming into the beam where these crosshairs are. And you can see them hopefully just getting totally destroyed and blowing up. So it's very satisfying to watch at the beam line. But this presents a unique problem in the sense that we need to come up with a way to continually prepare the sample in this S2 to S3 transition and bring it into the beam. And so we came up with this system that is being shown in this animation here, where we put the sample onto, this is a slurry of these little microcrystals that I showed earlier, put the sample onto a conveyor belt, move the conveyor belt through a system of laser flashes so that we can advance around that cycle that I showed earlier. And if we give it 2 laser flashes, it starts off in the S1 state because we specially prepare it so that it's purely S1. We give it 1 laser flash, it moves to S2, 2 laser flashes, it'll move to S3. And if you hit the sample with the X-rays, within microseconds of after the second laser flash, you will be able to catch it in this S2 to S3 transition. So that's what we're doing. This is what the conveyor belt looks like with a little bit more detail. We come in with the X-rays and we shoot the little droplets of sample right off the top of the conveyor belt. And we are able to simultaneously collect this X-ray diffraction signal and this X-ray emission signal at the same time so that we can monitor both the atomic positions and the electronic, or how many electrons are on the manganese atoms at a given time. This is what it looks like in reality. The initial experiment that we made in 2014 actually used real 1/4-inch audio tape, reel-to-reel, to do this conveyor belt system. We quickly abandoned that approach and went to this continuous belt that I showed you in the earlier slide. But still, these are complicated looking instruments, to say the least. And this is mainly the type of work that I was performing in this whole project. Okay, so now we can finally get into the data. This is another view of Photosystem II. It's kind of tilted. I'm not showing the membrane. I'm just showing the 2 monomers of that dimer. And this is the external surface of Photosystem II. If we start zooming in on it, you can see those coils and whatnot that I showed you earlier that stick into the membrane. Zoom in a little bit more. At the bottom here, I'm showing the oxygen evolving complex. Keep zooming in, take away some of the protein around it. And finally, we can get to this movie in the S2 to S3 transition. Okay, and so from this view, what we're looking at is the electron path out. So the electrons are going to be released from the oxygen evolving complex. They're leaving the manganese atoms, and they follow a sequence of molecules. First, the first molecule that picks up the electron from the OECs is being picked up by this red one here. And when it receives an electron, it kind of twitches. And by the time we get to the end of the movie, it is returned to its original state. So in the microsecond timescale here, we're looking at a movie that is a sequence of steps. It goes 50 microseconds, 150, 250, 400 microseconds, and then finally 200 milliseconds, which is much later in time. So only 6 frames that we're looking at linearly interpolated. very early on, it accepts this electron, and then it transports through the protein, eventually ending up on to this molecule on the left here, which is a quinone. And that guy also twitches and wiggles when it receives its electron. And finally, it'll end up on the right-hand side over here to this more violently moving quinone. And this quinone will actually take the electron away. So the reason this one's wiggling around so much compared to the other atoms is because it's on the surface of the protein and has much more freedom to move. So it receives its electron and then it will leave. The other thing to notice in this view is this cloud of blue balls. Those are water atoms. They're moving around a whole lot more on the right side than they are on the left side. Okay, and that's going to be important in a minute. So if we take on another view here, this is the same view or the same thing that we're looking at earlier, the same blue balls and so on. And here's that red molecule I was pointing out before. Now we're focusing in on this oxygen revolving complex. And I want to point out two more things here. If you wait, there it was. The most important thing is we see when we knew that there was going to be a water binding in S2 to S3 transition, and it pops into existence in this video here. Wait for it, there it is gone, and here it pops in. It happens around 250 microseconds into this transition from S2 to S3. So that's a very important observation. It tells us where the water binds. That's a key piece of information. You also see that the OEC is, it's wiggling around a little bit, but not too much, and it maybe elongates along this axis from the purple manganese atom in the back to the purple manganese atom in the foreground here. But it doesn't radically change shape. And that's significant because prior to measurements like this, people were simulating what was happening and using less direct measurements to try to see how this reaction would take place. And there were a number of people that were thinking that this structure of the OEC would kind of close in on itself. But that is actually not happening. And that's one of the advantages of being able to use a direct technique like this to look at where the atoms are. We can distinguish between these various theories more easily. Okay, so that was the last movie. Now that we have seen where the oxygen binds and we've seen the mobility of the waters around it, we can start to speculate how this oxygen formation reaction occurs. So there are three main routes that are favored in the literature at this point in time, based on this evidence that I just presented. And two of them think that the oxygen is formed between oxygen 5 and one of the adjacent water molecules. And then the role of this oxygen which just bound, this water that just bound, is to replenish the O5 oxygen here that I'm depicting after oxygen is formed, so that the structure remains intact or stable. And then the last suggestion is that O5 and the one that just bound, this OX, these are what form the O2 in the next step. And the role of the waters that are adjacent to it then replace this O5 to keep the cluster intact. We definitely favor these types of interactions because we also know that this calcium atom is important. And so somehow, if you replace this calcium atom with, for example, strontium, it will slow things down. So we know that whatever is attached to this calcium and whatever is close to this OX, these must be involved in the creation of O2. Another thing that's interesting and a really great example of why it's important to be able to see the entire structure all at once is the dynamics of the Photosystem II reaction. So what I'm showing here are these water channels on the bottom side of the protein. We have to get water into this OEC somehow. And we see in the structure that there are various channels of static water that lead towards the OEC, the oxygen evolving complex, this manganese center. And if you look at how those waters are moving around during this transition from S2 to S3, we find that this channel, the O1 channel, is much more mobile than these other two channels or several other channels. So what that means is these guys are banging around quite a bit during that transition. And we speculate then that because they're much more mobile, that this is where water comes in. Now, water doesn't leave this reaction, right? Because what comes out is oxygen gas and hydrogen ions. So the role of these static water channels is not to bring water in, but instead it's to transport hydrogen ions out. And that's how, this is a cartoon depicting how that happens. If you come in with a hydrogen ion and bump into one of these static waters, you can say, hey, can I take a seat? And it's like, sure, okay, but I gotta send one of my hydrogens over to the next guy. And he says, okay, but I need to send my hydrogen over to the next guy, and so on and so forth down the line until a hydrogen will pop off of this chain of waters. This works only if the waters are very still and arranged just correctly. And what's really interesting is we found that in this S2 to S3 transition, we have these still waters in various channels, but in particular, this channel here, this chlorine channel A, there's a residue here that rotates in position so that this channel opens or closes. During this transition from S2 to S3, the channel opens briefly, which should allow for one of these hydrogens to escape, and then it closes, which would prevent the hydrogen from coming back. This O4 channel, there's a water 20 here that actually disappears or becomes very unstable during this transition, so this channel is blocked. And this is really remarkable because what this means is the protein is involved in this reaction intimately, and it's basically got a little valve that's coordinating with whatever's going on in the oxygen evolving complex. And that explains partially why the protein sequence of Photosystem II, not just in the vicinity of the OEC, but also outside of it, is highly conserved throughout all these years. It's because this whole protein is involved in this intricate reaction. Now, I mentioned that we also use X-ray spectroscopy. I want to explain what its role is here. We used emission spectroscopy, and I showed this graph earlier that showed how when manganese atoms lose electrons, it causes the peak to shift. What this graph is showing is that during that reaction, we're watching how that peak shifts, and it's shifting by this according to this energy axis here. So by a fraction of an EV, electron volt, which is a unit of energy for X-rays. We flash it once and you flash it twice, and you can see the electrons leaving the OEC, leaving the manganese atoms in the OEC via this spectroscopy. You flash it 3 times. We start in S1, we go to S2, we go to S3, you flash it once more with 3 flashes, and you'll go from S3 through S4 to S0, and that's when the number of electrons return to the oxygen evolving complex. So that's what this is depicting here. The important thing is that in the crystals samples, compared to, say, protein solutions, that this reaction cycle, according to X-ray spectroscopy, looks the same. So we have this red, blue, and yellow lines that's here, and you can see all these other colors. They're all on top of each other, and they're all behaving the same. So what this tells us is that our crystalline sample is reacting and behaving the same as a solution of protein, which is very important because we want to make sure that the images that we're collecting with crystallography are true to the actual reaction. And the other thing that's important is look how consistent this is. So despite how complicated our setup is and how complicated this reaction is, experimentally, we can reproduce these trends. This is over several different beam times that each of these lines are collected. And the final thing that we really want out of X-ray spectroscopy is the ability to determine the causality of how things are happening. So for example, we want to know What came first? Did the oxygen get oxidized first? Or sorry, did the manganese get oxidized first? Did it lose its electron first and then the water binds? Or does the water bind and then the manganese is free to release its electron? That's not obvious. And if you have both measured at the same time and you're both time resolving both, there's a chance that we could see which of these events comes first. And so this graph is a little complicated, but these blue dots here, we're showing the progression of that manganese first moment. And you see it's going down because we're starting at S2 and we're going towards S3. So it's continuing down the curve. And it's doing so at a rate of around 250, 300 microseconds. That's the time constant. And at the same time, we're watching the occupancy of that OX that was appearing. And that's also rising at about the same time constant. And we're also comparing this to some other measurements from other groups, which are measuring also the manganese in different ways, or using either X-ray absorption spectroscopy, which I didn't explain, or IR absorption spectroscopy, which I also haven't explained, but these are complementary techniques that can all measure the same thing. And by and large, what we learned is, yes, we can see this reaction. It's behaving about at the timescale that we expect compared to other groups. But unfortunately, we haven't yet determined which came first, the chicken or the egg. Does the oxygen bind first and then the electron leaves the manganese, or vice versa? We can't really tell from this current data that we have, but I believe this technique is good. And if we get more data, we can start to answer some of these questions of causality. And there's lots of questions of causality in this situation. So I mentioned that there was this gate valve residue that was releasing the hydrogen ion. It would certainly be nice to really confirm whether or not this truly needs to release first and then let it out, or whatever. There's other residues near the OEC. So those are the questions that we have left. We want to know these causality types questions, and we would like to know, of course, what happens after the in the S3 to S0 transition. That is all, it's in the works, but it's not yet work that is finished. So I want to highlight the fact that I'm not the only person doing this. I've mentioned that many researchers have been involved in this over the decades, and indeed, even in this work that I just presented, it's a huge collaboration of some 50 people or more. Most of them are coming from Lawrence Berkeley Lab, but there's a number of people involved at SLAC. We have people coming from Sweden and Germany, and we've done this experiment in not just LCLS, but also in other X-ray free electron laser facilities around the world, for example, in Japan and Switzerland. It's really a big group effort. And what I also want to point out is that it's not just our group, this one that I'm discussing here, but there are other groups in the world that are studying the same reactions using slightly different techniques or similar techniques. And I think what's really encouraging is that there's a growing consensus and reproducibility in this kind of data that I'm presenting. So the story is converging, and we're coming to a much more clear understanding of this water oxidation reaction. So I think that's really encouraging. And I hope that you learned something. So now what I want to do is I want to take some questions, and I'm going to be helped in this question and answer session by two of my friends from Lawrence Berkeley Lab. We have Ruchira Chatterjee. She's an expert in many things, but among them is sample preparation of Photosystem II. So how those crystals are made, the magic that's involved in that kind of chemistry and purification, and how they're characterized by a variety of techniques. And then Aaron Brewster, who is the wizard behind producing these atomic structures from the crystallography data that I was talking about. All right. And thank you for your attention.
Okay, Franklin, thank you very much. This is really fascinating stuff. We have welcome to Ruchira and Aaron. And now we have time for questions. Many of you have put questions in the Q&A. And We're still accepting them, so you'll see a little Q&A box at the bottom of your Zoom screen. If you have a question, please write it in there, and we'll get it out on the floor. Let me start with a question from Gian. Now that we have animals, are we going to run out of oxygen? Is the equilibrium between oxygen and carbon dioxide reached yet, or what is the state of that?
Yeah, okay, that's an interesting question for sure. So I didn't mention in that geological record graph, the oxygen is not peaking right now. The oxygen peaked a while back. So when dinosaurs were roaming the earth, the oxygen levels were quite a bit higher. And I think, I'm not an expert in this area, but I think that is part of the reason why animals were allowed to get so big, is because there was even more oxygen and more energy available to people who were using aerobic reactions. So the question is it reached equilibrium yet? I don't think it's possible to reach equilibrium per se, because we keep changing how we're using oxygen. So humans, for example, are consuming oxygen much more rapidly than the animal kingdom was, because we use all these combustion machines. So I don't know if we're going to level off or it's going to keep decreasing, but it's less than it used to be some millions of years ago. But I wouldn't be too worried. I don't think we need to worry about a loss of oxygen in anytime soon. Carbon dioxide is more of the important gas to worry about.
Yeah, this oxygen, it's on a billion-year time scale, as I understand.
Yeah.
Jerry would like to know, are all plants using the same photosynthesis process, or are there differences? Or when you get to this level of the very core of the oxygen evolving system, it's the same in every plant, from a fern to a tulip?
Yeah, so photosynthesis is as I mentioned, a broad process, and there are different types of photosynthesis even in plants. I don't know, maybe you've heard of like C3 plants and C4 plants, and so all the structure that's involved in, for example, the chloroplasts, they're organized in different ways. Bacteria that do photosynthesis are much simpler in their structural organization of the internal bits, and that has an effect on how it is practically carried out. So in that sense, yes, there is differences between different photosynthetic organisms, but at the level that we've zoomed into here on Photosystem II, the point that I was trying to make was that Photosystem II is the same everywhere.
Oof. Very interesting. Stephen and Rebecca are both interested in the use of sunlight. So there are 4 photons that need to be absorbed in the process of Photosystem II as you go around that cycle. Are they all at the same wavelength, or each one is tuned to a different wavelength to do what it's supposed to do? And are there clues in that?
Sure, that's an interesting question that I studied quite a bit from my PhD time. So, right, photosystem, or the light is absorbed via chlorophylls mainly. There's a lot of light absorbing molecules going on in photosynthesis. And there are these antenna proteins that I didn't really talk about that float around outside of Photosystem II, and they can transmit light absorbed in the antenna protein to Photosystem II. And so by having these antennas that absorb light in different color wavelength ranges, you can get a pretty broad absorption spectrum. So it is able to accept light over a broad wavelength range. As long as it's above 680 nanometers, which is where the core of Photosystem II absorbs, that's pretty red light. As long as it's above that, then you can get it to work, because energy basically falls down a funnel and goes towards that 680 nanometer point where Photosystem II's reaction center functions at. There are some exotic samples using chlorophyll F. Normally it's chlorophyll A, and those systems are specialized photosystems that work deep in the ocean, where the only light that penetrates down there is deep red light. But the majority of plants, they use this 680 nanometers and above. That's where it absorbs.
So the absorption is nowhere near this oxygen transforming site. It's got its sensors way out there in the outside of the protein, and
Yeah,
gradually the energy is funneled in.
yeah, that's an important point because so manganese is a rare element on Earth, and in a plant per kilogram of plant material, there's only like 50 milligrams or so of manganese. So the plant doesn't want the photosystem II apparatus that I was showing to be the sole responsible actor in getting light. It'll produce much cheaper proteins that can absorb the light and then transmit it to the oxygen evolving centers. Of course, there's some trade-offs here. You lose energy every time you transfer. And so the organization of how the membranes are structured, that's actually pretty critical in how the plant is able to react to varying levels of light and so on. And that's where a lot of the differences in how photosynthetic yield changes from plant to plant and from different organism to organism.
Yeah, a couple of people wanted to ask you more generally about manganese.
Okay.
I guess Stephen, let me just read his question. Is the biological importance of early iron and manganese enzyme development based on their 1 electron or 2 electron preferences? So there are a bunch of questions wrapped up in here. I mean, the first one is, maybe you or Ruchir or Aaron would like to talk about the fact that there are metals in these things at all. I mean, ordinarily we talk about organic chemistry, and we're supposed to be made of organic chemistry, but then there are metal atoms inside these important structures, and different ones for different functions, like between photosynthesis and what's in our red cells, oxygen absorption. Can you say something about that? Why do these metals appear? What do they do? And why are there different ones?
That's a broad topic.
Sorry.
Why is it manganese and not iron, for example?
Is it just an accident, or is there a reason?
That's a great question. Ruchira, do you have any idea why it's not iron? I'm not 100%. I'm not 100% sure I know why it's not iron, and certainly it has a role. Manganese, for instance, has a capacity for much higher oxidation states than iron does, and that may be playing a role here.
I think it's redox potential driven.
Okay.
Could you explain that statement, please?
Because these are all oxidation processes, right? And this is one of the most oxidizing reactions known. So I think manganese was chosen from nature because it was capable to go to such high, like all 4 of them together could reach that high redox potential.
Oh, so it's not only even one metal. You need 4 metal atoms?
Yeah, there's
Yes.
4. Yeah, sure. Now, each of those manganese, though, we're pretty sure they don't exceed a oxidation state of greater than oxidation level 4, right? So manganese, maybe 5, yeah, maybe. Manganese can go all the way up to 7. That's one of its unique features, is that it's really capable of losing a lot of electrons before anything else. So I hope that answers some of the question that was asked. There's a lot of other questions buried in there about why different metals are used, but certainly the main role of transition metals like iron and copper and so on is to perform this redox chemistry, which means that they can store up the absence or excess of charge and then hold on to it for a while and then deliver it where it needs to go. It's like the capacitor of the protein. That's the role of the metal in this system, and in many of these systems. For example, also in hemoglobin, the purpose is to, well, actually, hemoglobin holds on to oxygen. Anyway, yeah, metals hold on to charge very well. That's what their role is in biology.
Interesting. So there are a bunch of questions about artificial photosynthesis. Ro would like to know, is it possible to accelerate the rate of photosynthesis? Either doing something to plants or maybe copying what's done and artificially synthesizing.
Yeah, okay. So Photosystem II, like I said, it can go up to 500 times a second. That's about the fastest you can get that system to recycle and do this reaction. Is it possible to go faster? Perhaps. For example, I mean, the most basic way to do this water oxidation reaction is to have a nice crystalline sheet of platinum and stick that into water and get it to react. But the reason by charging up with with electricity, that's doing the water oxidation reaction, as I was explaining in the electrolysis slide. Now, why doesn't nature? Obviously, nature doesn't have access to batteries, so it has to do what it has to do. But another thing that's important about the function of Photosystem II is its ability to control side reactions. If you dump enough energy into the system, you can get water to turn into oxygen like crazy, but you'll also start creating other things that aren't just hydrogen ions, electrons, and oxygen. And that's the danger. So part of the artistry of Photosystem II is to engineer not only the production of what you want and do that as quickly as possible, but also to prevent bad side products, because you're holding on to this, like Ruchira was saying, this it's powerful oxidant, and if left unprotected, that can just wreak havoc inside the cell and produce all sorts of bad things. So can we make it go faster in plants through bioengineering? Maybe, and that's certainly a thing that could be helped by understanding how it works better. But we do need to be careful that it doesn't start reducing the purity of the reaction that it's able to do.
Okay, very interesting. Xi has a related question. So she's interested in breaking down CO2 because of the global warming problem. And obviously, one way to do that is to plant a lot of forests. But is there an artificial way to do that's somehow suggested by our knowledge of photosynthesis?
Yeah, so there is, of course, a body of research in this type of thing. This is not what I really do.
Franklin, it's
yeah.
not what I really do either, but I do have a little comment on this, if it
Excellent.
helps.
Take it away.
What I can say, the name of this process is carbon sequestration, from what I understand. And it's a vital component of the Department of Energy's research portfolio for the next decade. It's like we have received directions at Lawrence Berkeley National Labs, which is where I'm at, to make this a priority, to prepare proposals on how to do this, to figure out a way to take the carbon dioxide out of the oxygen, out of the atmosphere, and sequester it into some kind of mineral. So one way people are looking at this is like the production of calcite, which is calcium carbonate. It's the material that seashells are made out of. And so there's research being done to try and do that artificially, perhaps using some kind of what's called a metal organic framework is what it's called. It's this lattice structure of metals and metals again showing up, right? But with to try and pull the oxygen down. It's active research. There are thresholds in efficiency that you have to pass to make it doable. Again, not my expertise, but a high priority in research right now.
I've got to ask you, do clams know how to sequester carbon?
I mean, that from what I understand, the calcium carbonate there is essentially sequestered carbon. Where they get it from? Do they pull it out of the ocean? I must assume so. Do they use it from their food sources? Maybe it's not not something I know much about.
Maybe we'll have another SLAC public lecture on that if we can find an expert. So let's see. Jim would like to know about the efficiency of this process. Whenever you design a machine, you're always interested in the efficiency with which you turn the eventual source of energy, in this case, red light, into your product. What's the quantum efficiency for photosynthesis? And is it remarkably high, or it's just there's a lot of light out there, so you can live with what you get?
The answer to that depends on where you want to draw the line for efficiency. So for my PhD work, we were focusing on the initial light absorption and how that's converted into an electronic charge. And there, the quantum efficiency is extremely high. So every photon that is absorbed, almost every photon, like 90% efficiency, results in the production of a charge. Now, as you progress down the chain, Photosystem II does a pretty good job of preserving for every photon. It does advance this reaction. But then once you start talking about how chemical energy is converted in the subsequent steps, obviously we lose a lot of energy in the release of oxygen. And then when you get finally to the production of sugar, there's a lot of losses along the whole chain there. So it's not perfect energy conversion all the way to sugar, but at least to the production of charge, it's very good.
Okay, Shantanu would like to know the following. Here you are with 4 manganese atoms in this huge superstructure of carbon, nitrogen, and oxygen. How do you find the signal? How do you keep the signals you're interested in from just getting lost in this enormous structure which you're trying to resolve?
Okay, that's a good point, and I'm afraid I didn't emphasize some aspects of that, of the answer to that enough. For example, with the X-ray emission spectroscopy, that is one of the unique advantages of it. It's, as I was showing in that graph, we have the emission from manganese is very well separated in energy from the emission of, say, iron or copper or carbon or oxygen, for example. And so that comes out, if you apply an energy filter, you can very easily see the manganese signal, no problem. Now, as for how the crystallography works, that it doesn't want to differentiate because you want to be able to see the carbon, oxygen, and all these different things. So it's collecting all of that simultaneously. And the only way that it's able to differentiate a carbon from an oxygen is due to the electron density that is inferred from the measurement. And this is probably where Aaron knows a lot more and can say more about how you can differentiate one element from another within the modeling framework that is used to analyze that data.
It's a really difficult question because, as you say, what you get from a protein crystallographic experiment is you don't, the blobs of density that you get and that you can look at, they're not colored in any way such that you would know what elements they derive from. If you see a ring, then it's probably a ring of carbons. If you see a blob near certain kinds of molecules, then maybe it's a water. And so we have to use other mechanisms to try and distinguish what are metals, what kind of metals they are. And yeah, sometimes we can do things like hit the crystal with a specific wavelength of light that we know will excite a kind of fluorescence, and then we can look to see if we see a change in this signal in the atom, and maybe that will help us. I did want to mention that protein crystals themselves are signal amplifiers, since that was part of the original question. And what they're amplifying is you take a single protein and you just make a million copies of it, all ordered the same way, and that amplifies signal.
Yeah, that's a great point. That's how those little spots, Bragg spots in the detector are formed is related to the fact that you have this crystalline lattice. It's a good point.
Okay, and then maybe to wrap up, there are a couple questions about practical applications.
Okay.
So are we learning something from this study of photosynthesis that will allow us to create fuels in a better way? I guess Stephen mentions liquid hydrogen, which is a fuel. Are there other fuels that we can imagine harnessing the photosynthetic cycle to make?
Yeah, so I think there's a couple lessons we can learn from this work. First of all, we've learned that in order so nature is very interested in doing two things. It wants to collect the chemical energy, and it wants to do it in a way that keeps the cell alive. So in that sense, it's very concerned with purity of the reaction. This is what makes enzymes so famous, is that they do precisely one thing and they don't do anything else. This is a problem with a lot of artificial catalysts where there's a bunch of side products that come off and you say, yeah, well, I got 80% yield. Well, that means that 20% wasn't what you wanted. And that's unacceptable for a cell because that 20% may be very damaging. So unfortunately, or not unfortunately, but industrially, it's not always necessary to have such high purity. So I guess what you can learn from Photosystem II is that that achieving purity requires a lot more effort. So that like gating mechanism that I mentioned earlier on the water channel and how it specifically allows the hydrogen out at one point in time and then closes the gate again, that's related to preserving the purity of the reaction. And I guess what you could learn is if you wanted that level of purity, you're going to need to design a system that has that level of complexity. And that's going to be very difficult. What was the rest of the question was, how can we, what sort of fuels can we make, right?
Does this help us make some kind of fuels? Maybe you say the criteria for fuel production is just different. And
yeah, I mean, you can use, there is research into using like bacteria to produce biofuels, of course, and that's possible. You do need to engineer the protein specifically for this, and Photosystem II is a way to provide cellular energy to the bacterium that's making. So for example, let's say you have a bacteria and you genetically engineer it so that it can absorb light and then produce it downstream. You want it to produce a fuel in that carbon fixation part of photosynthesis. So it's absorbing light, and then you specifically engineer it to produce a certain type of chemical that's easy to purify out that you can use as a fuel source. That's an approach that can work. Yeah, Ruchira, do you recall any good takes from JCAP times?
No, because as I say often, this protein is so complicated. I think the main thing they want to get is the principles in which it is working. If we can understand how it works, then they want to simplify it. Everybody wants a smaller, more efficient, cheaper materials, how to use iron, manganese to do the job. So the basic takeaway that I learn from any artificial thing is they want to learn the principles that nature is using, like how the proton-electron transfer and all that. But to replicate this, as you've already mentioned, it's very, very complicated.
okay, let's just take two more questions. First of all, there's a question from Jerry. Jerry must be a physicist. Can one use chlorophyll to make a photon detector by basically spreading photosynthetic stuff on a substrate?
I think there was some effort to use photosystem it wasn't really for a detector, but you can use Photosystem I, which is actually quite good and very robust and stable. You attach it to, I think it was gold, and this would, Photosystem I pumps electrons. So you can attach Photosystem I to these gold surfaces and use it to detect or to produce electrons, and that would produce charge. So yeah, I guess you could use it to detect things. I wouldn't say it's a very, the quantum efficiency of it, because of how you have to hook it up to something that's not biological, that might not be the best, but it could be possible.
Okay. And now the last question, maybe all of you can give us your own points of view on this. So now we've discovered that these photosynthetic processes are extremely complex and very specialized. And we've learned a lot about this particular one. Does it have more general lessons for biology, for what to expect, or other systems that we see in biology, or maybe other things that can be out there in the universe that we might encounter someday? What are we learning at that level from these studies of photosynthesis?
Well, from the perspective of X-ray science and instrumentation, we are certainly learning approaches that are successful for studying transition metals and redox reactions. So, for example, all this work that we put into studying how Photosystem II works has actually translated reasonably well to studying other redox active proteins. For example, ribonucleotide reductase, which is a protein involved in DNA synthesis. We looked at that, and that reaction wasn't light activated. We were activating it with oxygen gas. So that was an interesting application of the same kind of a technique and instrumentation, at least. What we've learned, I think again, one of the biggest takeaways from this whole deal is that when it comes to proteins, the mechanisms, usually you think of it like an active site, and that's true. That's a fine way to simplify how you think about it. But there's a reason why nature puts so much stuff around the protein, and so it's really important to be able to see the whole structure and to time resolve it, so that you can see what is the purpose of all this wrapper that comes around just the active site. Why can't nature just use, iron or something small wrapped around the thing? Why does it have to have all this stuff? And so I'd say one of the things that we're learning is that this is a great way to look at what is happening in the reaction. If you resolve it, you see these subtle changes in the protein. They can tell you a story about why there is all this stuff around it.
Ruchira, Aaron, do you have your take on this question?
Yeah, just expanding on what Franklin said. So as the first question asked, right, why manganese, why not iron? So as we are studying different metalloproteins, using this system, we have developed a lot of new instrumentation, which we are again using for different proteins, even MMO, which is methane monooxygenase, which so that's also very important metalloprotein out there. So I think the more we understand about the catalytic cycles of each of them, the more efficiently we can use it in mimicking and making, because everybody wants to make an artificial system. But since these are so complicated in nature, we have to we are right now using Photosystem II to build our instrumentation and then study the rest of the proteins.
Aaron, Franklin, any last words?
Well, I was going to comment that I really liked Ruchira's early answer about the complexity of the system, right? Being able to look at gigantic structures as a whole and get all this detail is just so valuable towards building new tools to help resolve the environmental problems that we discussed. But I also wanted to mention that through this project, we're learning a ton about the physics of X-ray diffraction inside the crystal itself, which is really interesting on its own. Just how crystals grow and how crystals pack and how they diffract and what changes you get in the images and how can we really resolve that data more carefully. It leads to more detail in not just this project, but many of the other projects that we collaborate on. So there's a lot of really interesting methods development that we're doing, which is really fun.
Okay. Before I end this and thank you guys, let me just tell the audience that when this thing ends, the poll will appear on your screen. So please hang around to take the poll. It'll be very short and help us with the structure of these public lectures. So now I'd like to thank Aaron, Ruchira, and especially Franklin for this very informative talk. And we're very grateful, and we hope to see you folks, the audience, at the end of January for the next of these public lectures on a very different subject, the magnetism at the center of the Earth. And until then, enjoy yourselves and stay curious. Thank you very much.
Thank you.
Thank you.